thank you, all of you, for coming, despite dissertations and review essays and all the other things we have to do. So this is the Psychology Forum, and this year the theme is Research Matters. And I won't go on very long, because we have short time. But I will say that at 2 o'clock, we're going for refreshments in the foyer. And about 2:30, we'll gather here again for a panel discussion about the topic of impact, and you have the opportunity to ask questions or give comments on what you think. And yeah, I'll just leave it at that and introduce this first speaker, which is Professor Keith Siller, who is going to talk about who cares about fish and frogs, the value of pure research.
Thank you very much. Thank you for coming, everyone. Thanks for the opportunity to address a very different audience for me, having just come from biology to psychology and neuroscience. I'd like to say that I've probably been given the greatest challenge, which is to persuade you, if I can, why any research on animals as simple as fish and frogs might have any relevance for science and for society. So before I came here, I was chatting to my 85-year-old father and explaining to him what I was about to do. And my father is always very polite, but I also know exactly what he's thinking. And this is the kind of thing that he might have been thinking. So my son's a professor, a grown man, and he studies tadpoles. And then he would never say it, but he might think, some of us really had to work for a living. So I have to try and justify to you why it's relevant for a grown man or for anybody to study simple systems. I'm going to be talking about neuroscience research rather than psychology, obviously. And I think the real justification is simply that we are human beings and we are very inquisitive creatures. And I think that we have an intrinsic thirst for knowledge, for understanding things that we do not understand, and for asking very simple questions about how things work. This is part of the human spirit. And I think that's justification enough for doing blue skies research, simply to create new knowledge that can be passed on from one generation to the next. The fact of the matter is that society wants everything that we do to have a value for society. But in my view, not all research has to be translational. And the reason for that is that the outcomes and benefits of basic research cannot be predicted. So what I'm saying is that knowledge, for knowledge's sake, is strength. Now, one of the ways that pure research can benefit mankind is simply by observing what happens in the world around us. So here's an example in which the design of a bird can influence the design of an aircraft in order to save fuel. But of course, we're part of a School of Psychology and Neuroscience department, so we need to ask, what is it about the design of nervous systems that animals can tell us about ourselves? Well, I thought by analogy I'd start by showing the pinnacle of automobile evolution, possibly. The Ferrari Enzo. It's slick, elegant, it's got a lot of horsepower under the bonnet. It's very impressive, but it also has its pitfalls. It's slightly temperamental. It's high maintenance. It needs a lot of servicing. And in a way, human beings fall into the category of the Ferrari Enzo. But if you were to be parachuted in from outer space and asked the question, go and figure out how the internal combustion engine works, then I would propose that you'd be far better to study something like a Fiat Cinquecento, because it has the same basic principles of how it works, but getting at how it works is much easier. So the equivalent of Steve is the laboratory mouse, but even mice are very complicated, and so I would argue that by studying even simpler vertebrates, like fish or frogs or frog tadpoles, can give us insights about how the nervous system actually generates behaviour. Now, it's a startling fact that about 90% of presentations at the annual Society for Neuroscience meeting are on mice, and almost all of those are on some form of transgenic animal. And that's very important because, of course, mice can tell us a lot of information that's relevant to biomedical research. But I would argue that by taking a very small snapshot of Noah's Ark, of the designs of animals that are out there, is missing a lot of basic information. So I would make a very strong plug that not only is basic research important, but that comparative basic research is also very important. Well, I study animals at a very early stage of development, and my justification for doing that is that they have simple nervous systems that one can study at the level of single neurons. But does that have any relevance for human beings? Well, many of you will recognize this image. You may not recognize the person. This is Ernst Haeckel, who came up with the very erroneous suggestion that during development, humans follow and recapitulate their phylogenetic origins. So we start, first of all, in the womb as an embryo that's basically a fish, and then that progresses up the evolutionary tree until we finally become a human. Well, he's completely wrong, but the images are very compelling because at very early stages in development, you would be hard pushed to distinguish any of these vertebrate embryos from any of the others. And it's only as development proceeds that the developmental expression of genes transforms these embryos into the animals that we recognize as fish, pigs, humans. But if you go back to the earliest stages, of course, the embryos are all virtually the same. And they're the same because of common ancestry. And what that tells us is that there are common underlying mechanisms that control their development and the activity of their brains. Well, this is what I justify to myself anyway. So has any of the work on simple systems been of any value? Well, I would say that most of the general principles of neural system function have derived from Blue Sky's research on simple animals. Here are just a few. The ionic basis of the action potential, Hodgkin and Huxley. Role of calcium in synaptic transmission, Rodolfo Linas. Synaptic excitation and the role of acetylcholine, Bernhard Katz. Synaptic inhibition, Steve Kufler. Role of NMDA receptors in LTP, Richard Morris from St Andrews, it was discovered. So there are general principles that have derived from work on simple model animals. The reason for showing you Groucho Marx is he famously once said, "I have my principles, you know, but if you don't like them, I have others." There are other tangible benefits of pure research. Is it translatable ever, even if you didn't intend it to be in the first place? I'd like to show you an example from work that was done in St Andrews about 10 years ago by a character called Nick Dale. And Nick Dale was studying the control of activity underlying swimming in a frog tadpole. And he discovered that there is a very elegant biochemical pathway that regulates the excitability of the spinal cord, in which during activity neurons release ATP that causes excitation, but with a delay enzymes break down the ATP to adenosine that has the opposite effect. So there's a built-in negative feedback control system to make sure that eventually the activity stops. To prove this little simple model, Nick had to design probes that allowed him to measure the concentrations of ATP and adenosine. It turns out that those levels fluctuate dramatically in a variety of brain injuries and brain pathologies, such as stroke. So Nick took the design of his simple probes and he created a spin-out company called Sarissa, and they sell to clinicians devices to measure levels of adenosine in human brain. So who'd have thought that work on locomotion in a tadpole could actually make it into the clinic? Another value of pure research is that simple animals tend to be able to do things that complex creatures like Steve are able to do. And one of those is spinal cord regeneration. We, of course, as humans, are very bad at regenerating axons in the nervous system, but simple animals like fish, salamanders, frog tadpoles can all regenerate neuronal axons. And here's an example from a zebrafish. This is actually a single neuron located in the brain that allows the fish to escape from predators. If you cut the axon of that neuron, over time it will grow back into the spinal cord. It's not the only neuron in the fish that does that. If you take an adult zebrafish and do a complete spinal transection, 6 weeks later, it's behaviourally back to normal. Back to normal because axons can grow across the crush site, which simply cannot happen in humans. So, zebrafish probably hold the key to unlocking the secret of how we might be able to regenerate nerve cells in our brains. The final point I want to make, which maybe relates more to psychology than anything I've said so far, is the fact that certain simple model animals may give us insight into how we might bridge the gap between knowledge at the level of individual neurons and complex behaviour, maybe including cognition. This is a recent paper from this year, I think, on zebrafish, and these are becoming a fantastic model for understanding brain circuits. This little zebrafish is about 3 millimeters long. It has a completely transparent body, and you can see right into its brain. Its genome has been sequenced, and in this particular transgenic fish, neurons fluoresce when they're active. And you can image the living brain of a zebrafish held in position using 2-photon confocal microscopy. And you can then present the animal with some kind of behavioural task and ask, at the level of individual nerve cells, which neurons are active during that task. And these images on the bottom show the entire brain. These are individual neurons and nuclei that are active during locomotion and during the presentation of a sensory stimulus. So for the first time, I think, it's now feasible to look into the brain of an animal and at the level of single cells ask which neurons are active when the animal is learning or changing its behaviour. So I hope I haven't gone over my 15 minutes, and I hope I haven't insulted the person who invited me to speak here, and leave it at that. Thank you very much.